Okay, so Assalamu alaikum everyone. I'm Momo Arai. I'm a fifth year medical student, and today I'll be talking to you all about the urea cycle. Now, if you have, I know this is a recording, but if you have any questions throughout or uh, try to collect the questions, and you can ask me anytime, actually, anything related to the lecture or not, related to MOL, anything like that. You can reach out to me through my email if you have any questions, okay? And yeah, don't hesitate to ask me anything. Now, I'll get started with the cycle. So, inshallah, it's easy and inshallah, it goes smoothly. This cycle actually is, for me, was a lot easier than all the other cycles you might have seen in MOL, uh, just because the enzymes that you will see are related to the names of the cycle. You'll see what I'm talking about. And once you know the cycle, like once you memorize that cycle using your mnemonic, using the pictures, you'll be able to better understand and tackle the questions that come for this. Okay, so inshallah it's easy. Again, let me know if you have any questions through my email. Now, I'll just start to I'll just want to start to talk. I just want to start to talk about the urea cycle. What is it and why do we need it exactly? So urea cycle, as you can see from this picture, um, it takes place in the liver. Okay, that's one of the main points that I want you to get out of this lecture. What is it exactly? Why do we need it? Okay, so from the name, it's urea cycle, right? So it makes urea as a product. Um, but why do we need it in the first place? Okay, so all of us, when we eat proteins, any food that has high protein amounts and anything like that, we will have proteins in our body. Now, proteins are just one of those substances that can't be stored, like fats or anything like that. So, protein, if someone takes too much protein, if they have excess protein, uh, excess amino acid, it means the same thing. So, excess protein, excess amino acid, excess nitrogen, all of these mean the same thing. If someone has excess of them from eating from their diet, this protein in their body needs to be broken down, right? It will be broken down through a process called deamination. This is another lecture you might have taken. So this protein, excess one, will be broken down into by deamination into something called ammonia. Okay. Now, ammonia is a toxic substance, and you don't want this to be around in your body. You need a safe way for ammonia to be excreted out of the body. And that is the purpose of the urea cycle. So the urea cycle takes this toxic ammonia. So wait, let's start from the beginning. So excess nitrogen, amino acids, excess proteins converts or breaks down into ammonia. Ammonia is toxic. So this urea cycle takes the ammonia, this toxic ammonia, converts it to urea, and urea is safely excreted out of the body. And this urea cycle takes place in the liver. Okay, you will see here, it converts ammonia into urea, just like this process is showing you. Now, I hope that's clear. This is why we have the urea cycle in the first place. Now, I mentioned that ammonia is toxic, right? And you don't want it to be around in your body in the first place. So how does ammonia get to the liver? So all your cells have all this excess protein. How does this go to the liver in a safe way? So that actually happens in the form of another safe amino acid called glutamine. Okay. From this slide, just know that glutamine is the non-toxic carrier of ammonia. So why is that? Again, ammonia is toxic. You don't want that to be around. So in these cells, in the muscle cells, wherever you have excess amino acids or proteins or excess um, nitrogen that gets broken down into ammonia, this ammonia needs a safe way to travel from these cells to the liver so that the urea cycle can take place. Okay, how does it do that? So ammonia, this one in other cells, will combine with glutamate, okay, with the enzyme. So, okay, ammonia will combine with glutamate to make glutamine. Through the enzyme from its name, glutamine, making glutamine. So glutamine synthetase. Okay. So again, ammonia is made from deamination of excess proteins, amino acids in most tissues, and is transported safely by as glutamine here. So glutamine is very, very safe. It will go through the blood and all the way to the liver. Okay. Again, like I mentioned, this is the enzyme name, glutamine synthetase, combining glutamate with ammonia in other tissues. And then glutamine is safely carried to the liver. Now you will see this one, or kidney. It's just because some of the urea cycle can take place in the kidney, but it's not the most important like site of this cycle. The most important place where urea cycle takes place is the liver. Okay, some can happen in the kidney. That's why it's written here. Okay. Now glutamine is safely transported to the liver, right? Then it breaks down into its original substrates, like how did it form in the first place? So glutamine will be broken down into ammonia again, which can be picked up by the urea cycle now because we are in the liver. And the other product is glutamate, which we started with originally. So in the liver, it's broken down into ammonia, back into ammonia and glutamate. Glutamate undergoes other reactions called transamination reaction. Basically, it can change to other forms of amino acids. This is another lecture on its own. You might have seen this somewhere. So glutamate converts into alpha ketoglutarate. So glutamate, it's the same as glutarate. You can memorize it like that. And other sorts of or forms of amino acid like alanine, and again, pyruvate. So this transamination reaction is another lecture on its own. Just remember that glutamate or these amino acids have the ability to uh, become other amino acids, and even glucose can be related to that. So don't worry about this for now. It's not the focus of this lecture. And even this one, glucose alanine cycle, it's another lecture on its own. Okay. But the whole point of these cycles are they're all interconnected in some way or another, right? Like this one. But it's not the main focus of this lecture. So yeah, I would just stop here for this one. Glutamine is safely transported to the liver, broken down into ammonia, which is carried into the urea cycle, which I'll cover right now, and broken into glutamate. And glutamate will undergo all these other reactions. Okay. Yeah. So I hope that is clear so far. So just remember these names, this red one, glutamine. Okay. Okay. Now this is the whole point of this lecture. So if you have excess proteins, amino acids, things like that in your body, it will be broken down into ammonia. Now ammonia, once it's safely transported to the liver through the glutamine, and it becomes ammonia in the liver, it will undergo the urea cycle here. Okay. Urea will go to the kidney to be safely excreted now. Okay. Because it's a safe substance. So this is the whole lecture kind of summarized, or whole idea of the lecture. Now, okay, I'll start with the urea cycle. This is the whole cycle. Okay. If you remember, again, you can use any diagrams for you to memorize whatever helps you memorize it. Use that cycle. You'll see a lot of it through the lecture, but this is a nice one, and it's a kind of a nice summary for the cycle. I'll go over this one, but before that, I just want to talk to you about urea, the structure of urea. So this small structure you see here is urea. You will notice that it has two nitrogen containing groups, one here, and one carbon containing, right? So there's one carbon in between. Where do these come from? In general, you will see during the cycle what I mean, but uh, because urea cycle, we said it converts ammonia to urea. That's the whole point of the urea cycle. One of these nitrogen will obviously come from ammonia or ammonium ion. So the difference between this and this is just the ion, like plus sign. It's the same substance at the end of the day. So ammonium ion or ammonia gives you one nitrogen. Okay, one nitrogen atom comes from this. What about the second nitrogen in urea? It comes from an amino acid called aspartate. Now here, one nitrogen comes from aspartate. Aspartate, you will see it in the cycle later on. It's important because it enters the cycle to give you the second nitrogen in urea. Okay, so those two are down, one nitrogen, one nitrogen. One carbon, this one, the one in the middle, comes from carbon dioxide. So carbon, carbon dioxide. Um, that's just where it comes from. Sometimes you will see later on, you will see this as bicarb. It's the same thing. Bicarb or bicarbonate ion is just the dissolved form of this carbon dioxide. But yeah, it's the same thing. You could write slash here, bicarb, giving you carbon. Okay? Okay. We covered that it takes place mostly in the liver, this whole urea cycle. The last point, so I covered the structure of urea, where each structure comes from, and now I'll go into the cycle. Now for this slide, I will say it in general, and then we'll go more into detail. So don't be like, you don't need to be overwhelmed. And I'll of course explain the rest in the coming slides, okay? Okay, so the first reaction that takes place is the formation of the first substance, which is something called carbamyl phosphate. So this, what I'm drawing here, this like box, is the first step of the urea cycle to make carbamyl phosphate. Okay, uh, carbamyl phosphate needs substances to make it, such as carbon dioxide and ammonium. So we covered these two down in the first step of the reaction to make something called carbamyl phosphate. Now, second step of the reaction, carbamyl phosphate reacts with something that is already found in our cells called ornithine. Okay, so ornithine, carbamyl phosphate combines together to form something called citrulline. This is the second step of the reaction. Okay, second step of the cycle. Now, because ornithine is already there, okay, so it just gets used up in the cycles as I'll show you now. So, okay, second step, carbamyl phosphate, the first product, combines with ornithine to make citrulline. So that is the second step. Citrulline then goes on to combine with aspartate. So that's the third nitrogen atom that's remaining from these, right? So we're hitting all these points now. So citrulline combines with aspartate to give you a long molecule called arginosuccinate. Arginosuccinate, since it's very big, it will break down into fumarate and arginine. Okay, arginine combines with water in a hydrolysis reaction. This is the last step, making urea, our product in the last step, and ornithine. Now, ornithine, you realize it's the first, it was used up in the second reaction in the urea cycle. So that's what I mean. The last reaction makes ornithine, and we said the second reaction uses up ornithine. So ornithine is being recycled again and again. Okay, so that's why it acts as a carrier for all of these molecules to make urea. That's what this sentence means, okay? And again, like I said, the first reaction is this one, formation of the blue arrow. Second reaction is this one. And then third is this one, and so on. Okay, I hope that's clear. Again, this is in general. You will see, this is another cycle for the urea cycle. It's very complicated. It was just in your lecture, so I included it here. But just remember that urea it is made up of two nitrogen, one coming from ammonia or ammonium, and one coming from aspartate. And the one carbon is coming from carbon dioxide. Okay? Or, like I said, bicarb, as you will see later. Okay, so these are again other cycles. Just choose one to memorize. Don't confuse yourself with a lot of cycles. It's okay. This one is a very, very complete one. This is I used it from like a YouTube called Dirty Medicine. He summarizes it very well. That's why I included it. For this one, just you can focus on the purple thing. So as you know, all cycles, all reactions in MOL, they have an important step, okay, which is called the rate limiting reaction or rate limiting step. That actually, for the urea cycle, it's the first step. So I'll use this one because I already explained here. This step, the first step, is the rate limiting reaction. So that's in purple here. This is the first step. It's just flipped the cycle. So that I don't want to confuse you too much. Just follow one cycle for you to memorize, okay? And this is just showing you that urea, after you finish the cycle, goes to the kidneys to be excreted. Okay, so this is a mnemonic for you to memorize the substances that were involved that I just said. And I correlated it with this diagram. This diagram is showing you, so the molecules in orange, the boxes, plus the red ones are what is included in the mnemonic, okay? So the mnemonic goes, you can use any other mnemonic. This is just the one that's like very common, that's why. So orange colored cats always ask for awesome umbrellas. So that's the mnemonic. Now, each of these words correlate to, again, like I said, it starts with O, right? Orange. So what's the only O in this cycle? Ornithine. Ornithine is the carrier that keeps going again and again in the cycle. So that's what we start with, ornithine. Okay. Then colored is carbonyl phosphate, right? This is the first step of the reaction or the urea cycle. So ornithine is the carrier. This is the first step. They both combine together to make something called citrulline. Now that's cats. So orange, ornithine, colored, carbonyl phosphate, cats is citrulline. Always is here. So citrulline combines with aspartate. You remember this one? Citrulline combines with aspartate to make this long molecule. So okay. Citrulline combines with aspartate to make arginosuccinate, which is this one. Always is aspartate. Asp is arginosuccinate. Arginosuccinate breaks down, it's a big molecule, to two things. So 4 is for fumarate, and awesome is for arginine. Now, arginine, if you remember the last step of the reaction, arginine combines with water and gives you urea, which is the red box. So here, umbrellas. Okay, that's the last step, is the product that we all want, or we are all talking about. And the other product is ornithine. We said ornithine keeps going back again, so we're back to orange. Okay, that's why that's the first one anyway in the one. So okay, again, so orange colored cats always ask for awesome umbrellas. So wait, I'll try to go here. Yeah. So this is the first cycle that I explained. So orange, ornithine, orange colored cats always ask for awesome umbrellas. Do you see? So this is very, very much more clear actually, because you don't have, these distracting you in the middle. But yeah, you will see how the mnemonic fits right in with this cycle. Again, use any cycle that is better for you to memorize. And don't worry too much as long as you cover the whole uh, substances, okay? All of them. Now, as we said, the urea cycle happens or takes place in the liver, right? That's the first point. Now, within the liver, you have two places where the urea cycle takes place. Part of it takes place in the mitochondria or mitochondrial matrix, and the rest of it takes place in the cytosol or cytoplasm. Um, so that's why it says compartmentalization. Okay, so it takes place in two different places in the liver itself. The first two reactions, this one, making of carbonyl phosphate and making of citrulline, happens in the mitochondria. So we start small and then we go big. So this is in the small organelle, and then citrulline, so C, after this is formed, C goes to the cytoplasm to continue the rest of the cycle. So the rest of it, like you can think of it like the bigger part of the reaction happens in the cytosol. Okay. I'll explain now each step by step. So the first step, like we mentioned, is the blue here, this one, making of carbonyl phosphate. And you will see the small icon. I just put it here to tell you that it takes place in the mitochondria or mitochondrial matrix, to be specific. Now, okay, three things that I want you to remember. Formation of the carbonyl phosphate is the first step of the urea cycle, made from three things. Two of them we already covered. So one, ammonium or ammonia, like I said. And one here, see, it's written bicarb or carbon dioxide. You will see in other places. So one bicarbonate ion, one ammonium ion, and the last thing is energy. So two ATP molecules, meaning energy is used for this reaction. Okay, so these three things make carbamyl phosphate. What is the enzyme? From the name, carbamyl phosphate synthetase 1. Because it's the first step, it's 1 in the urea cycle, and synthetase because it makes this carbamyl phosphate. That's why I meant the enzymes are easier to memorize now because the names are very like similar, right? Okay. The other thing that I want you to know about this is that it is the rate limiting reaction, okay? Rate limiting reaction, again, it's the most important reaction in the whole cycle. It determines whether the cycle can, take place um, in at how much rate, I guess. So either fast or slow. It controls the whole cycle, basically. It's like the controller of the cycle. Now, this this also means that this step is regulated, can be regulated by other things. What is that thing? It's called something called N-acetyl glutamate. Okay, so N-acetyl glutamate is regulating this step, the rate limiting reaction. Not only regulating it, actually, you actually need this for this to happen. So that's why it's called an obligate activator. You really need this to start the cycle. If you don't have this, if someone doesn't have this first step, the most important step will not take place in the first place, right? So this is why it's called an obligate activator. Remember that for your exams. And N-acetyl glutamate itself comes from other amino acids. So that's why it is produced after protein-rich meals. We will get into this later, don't worry. So by this we covered the first step of the urea cycle. Now, the second step of the urea cycle, we said after forming carbamyl phosphate, which is this one, it combines with the carrier called ornithine to make citrulline. What is the enzyme for this? And oh, okay, before that, it takes place again in the mitochondrial matrix because we're in step 2 of the urea cycle. So it ornithine combines with carbamyl phosphate made from the first reaction to make citrulline. What's the enzyme? Again, because ornithine is involved, this is now called ornithine transcarbamylase or OTC. Okay. Okay, remember how I said the first two happen in the mitochondria? So after this, after citrulline is formed, citrulline goes to the cytoplasm or cytosol to continue the rest of the reactions. Now, this is more in detail. Oh, wait, sorry, this one, yeah. So you will see that ornithine combines with carbamyl phosphate to make citrulline, as we just said. This enzyme helps in doing so. From the name, ornithine is ornithine. Transcarbamylase means like transferring a group, right? If you think about it. So what is it transferring actually is this phosphate group. So if this phosphate group is released in this reaction, and you will see that that's how this part attaches to the head of this to make one substance called citrulline. Okay, so in this process, in this reaction, you have release of high energy phosphate. So high energy, this is a high energy bond. If you release phosphate, that means you are releasing energy as well, because phosphate groups they have a very high energy within that bond. Okay. Okay, and this is what you can see from the picture here. As you know, we said that urea is made up of 2 nitrogen, 1 carbon. We already covered the first two. So 1 nitrogen we covered from ammonia or ammonium, right, from this one. And 1 carbon we covered from the bicarb or carbon dioxide, right? So we covered 2 out of the 3 things that we need for urea already. That's what they mean. So this step makes citrulline, which combines 1 carbon and 1 nitrogen into one, you can say, substance, which will later become urea in the following steps. So the first 2 are down. We have 1 more to go. 1 more nitrogen to go, actually. Okay, so the third step of the urea cycle is something called So, okay, wait, this is the enzyme name. So the third step, we said citrulline goes to the cytosol or cytoplasm to continue the rest. Citrulline combines with, yeah, this is the one, aspartate. Now, aspartate is the third nitrogen that is needed for urea to form, right? So citrulline, third thing, so this is the third step. Citrulline combines with aspartate to make arginosuccinate. So I'm sorry if like I'm confusing you between the cycle. They're all the same thing. So like for example here I cut off like I keep I think this was in the lecture anyway, but I kept cutting off like this is the first step, then you continue second, and then this is the third step. So citrulline goes to the cytoplasm and combines with aspartate and energy, ATP, even here, to make arginosuccinate. Arginosuccinate is a very, very long molecule. See, it combines everything. So that's why it needs, you can say energy. Another way I like to memorize this is the enzyme names. So this one is called arginosuccinate synthetase, making of arginosuccinate, and that uses energy. Synthetase, whenever you see that in this cycle, it needs energy, okay? The first, for example, here, carbamyl phosphate synthetase 1 needs 2 energy molecules, 2 ATP. So I just like to know that synthetase needs energy. It will help you for the urea cycle. The second one is ornithine transcarbamylase. We just said it releases phosphate by the word trans. And then this one, synthetase, is making arginosuccinate using energy, one more ATP molecule. Okay, so yeah, so it catalyzes this reaction, and we introduce the last nitrogen atom that we need for urea through this molecule, okay, through aspartate. Okay, now the fourth reaction you see here, after it forms arginosuccinate, I said it's a very, very big molecule. It will be broken down into arginine and fumarate. Okay, fumarate will leave the cycle. It's okay, I'll tell you where it goes later. So it leaves the cycle, and arginine continues to the next step. Now, the enzyme that catalyzes this reaction is called arginosuccinase. So arginosuccinase breaks down arginosuccinate into 2 molecules. And here, okay, this is, again, it's like a side note, but it just says it completes the transfer of amino group from aspartate, which is this one, to arginine. Just in easier terms, it just means that it makes arginine, okay? And that nitrogen from the aspartate group is embedded in arginine. And again, here, you see how this was aspartate, right? The nitrogen goes and is embedded on arginine, the next step. And you see how this skeleton is the same as fumarate? Like it's just missing that one nitrogen. That's what they mean by the last point. I'm just trying to explain. It's a very minor thing. So it says retains the carbon skeleton of aspartate as fumarate molecule. Okay, I'll just like start from the third. This is the third step. We're now back to the third step. We said citrulline combines with aspartate to make a big molecule, arginosuccinate, through this enzyme, arginosuccinate synthetase. Fourth thing is that arginosuccinate breaks down by arginosuccinase enzyme into arginine and fumarate. So basically this aspartate just gave its nitrogen to the arginine, and it's fumarate looks like aspartate basically without the nitrogen because arginine took it, okay? That's what they mean here. Okay, so that this is the third one, synthetase, breaking down is fourth step. Now we have arginine, right? This will continue in the fifth step and the final step of the urea cycle. So arginine combines with water in a hydrolysis reaction through the enzyme, again, arginase. So it breaks down arginine into 2 things again, okay? Which is ornithine and urea. Like we said, this is the final step of the reaction. So we will have our product urea with the 2 nitrogen and 1 carb. So this point is carbon. So 1 carbon, 2 nitrogen. That is urea. What is the other product that is formed? Ornithine. Ornithine is the carrier that goes round and round, right? So it goes back up the cycle, combines in the second step to form citrulline. So ornithine just goes back up to the cycle and repeats itself. Urea is the product from this cycle. So, okay, so arginase catalyzes hydrolysis of arginine to ornithine and urea, and ornithine cycles back to the first step. Actually, it should be called second step because the first step is this one, but you get the idea. It just cycles back to go to the beginning. Okay, picks up another carbonyl. Yeah, that's it. Okay, so we covered the urea cycle, we covered the enzymes, we said the first is the rate limiting step. It's regulated by an acetyl glutamate to form the first substrate. This substrate combines with ornithine, the recycling thing, to make citrulline. Citrulline goes to the cytoplasm, don't forget that step, and combines with aspartate to make arginosuccinate through this synthase enzyme. Arginosuccinate breaks down into arginine and fumarate. Arginine continues and breaks down again by arginase to ornithine and urea. Okay, I hope that is all clear. So that is the overall cycle of urea cycle. And okay, all of this honestly, it says the same thing that we mentioned before in just in word for like format, like in terms like in words. And this is the overall reaction you can say if you put it all together. So this and this, same thing. So you see how it starts off with carbon dioxide, ammonium, these two things needed for urea, and ATP. Now, ATP, we said, oh, here. So we said in the beginning it needs 2 ATP, the synthase 1 enzyme, 2 ATP here, and 1 ATP in the other synthase enzyme, so ATP here. So in total, you will use 3 ATP for urea cycle, okay? And that's why it's 3 here. We use aspartate. That just means that aspartate enters the cycle. And water molecule for the last step, which is hydrolysis. This will form urea, which is the product, plus okay, so I'll talk about this part in a bit, plus fumarate, which leaves the cycle in the middle, right? We said fumarate leaves it here. Okay, so these 3 ATP molecules, two of them, okay, here, if you see this one, okay, I'm just trying to go to the first step. Oh, okay, they didn't mention it, sorry. So yeah, here, it's here. The first step, the 2 ATP that we used in the first step, it actually gets converted to or breaks down into 2 ADP plus phosphate, right? So each, you can say, I can draw it if it's more clear for everyone because I'm not sure. I really hope everyone like gets this point. If you do get it, you can like skip this part, it's okay. So ATP is like it has 3, you can say, oh, sorry, so 3 phosphate molecules, right? 1, 2, 3. Now, again, if you break one bond, it's going to be ADP and one phosphate, right? So that means ADP means having 2 phosphate plus this broken bond becomes the phosphate alone, okay? Or you can have, if you have 2 broken down, 2 phosphate bonds that breaks down, you will have AMP, which means it's only left with one more. Okay, AMP is one more phosphate plus PPI. So that is like you can say phosphate, 2 phosphates go here. Well, I'll try to explain again if you didn't get it. Okay. So ATP has 3 phosphates attached to it. So 1, 2, 3. If one phosphate is broken down, it will be left with 2 phosphate groups, and that one phosphate will go out like this. So it was 3, it became 2 plus 1 phosphate alone. Now, if you break 2 bonds here, this all happens due to the reaction. If 2 bonds are broken down, this ATP, it was 3, So 2 is gone, so it's left with now 1 phosphate bond to the AMP, and it's called AMP. And the other 2 that is broken down will be called now PP. So phosphate, 2 phosphate groups basically are separated. So that's why you will see the first reaction that used ATP converted into the first thing, ADP plus phosphate. Okay, so only one bond was broken here, one bond. The other synthetase enzyme that used ATP was broken down into AMP and 2-phosphate. So in this reaction, 2 bonds were broken and it created AMP plus, so monophosphate, plus the 2-phosphate that was broken here. So the 2 is for this reaction. If one bond is broken, it gives you ADP, diphosphate, plus the 1-phosphate that goes out. I hope that is clear. I hope that makes it clear. It will help you in other cycles as well if you see this. So ATP can be broken down into 2 ways. It can either form this, ADP plus 1P, or AMP plus 2P, depending on how many bonds were broken. Okay, now okay, so in the urea cycle, we covered all of this, right? So we said 2 ADP plus 2 PI from here. This should be a, yeah, here it says overall, okay? Because you have it here. So 2 ADP and then 2 phosphate alone plus 1 AMP plus phosphate together, so PPI alone. Now, again, this is more like I'm going into the tech, like the small details, so don't worry about it. If you understand it, just you don't have to like know this part as much. I'm just trying to make it clear in case anyone is confused. Okay, so this is the overall reaction. Now here it's the same thing. They just left out this and aspartate and fumarate just because they enter and leave the cycle. It's not important in the overall cycle. That's fine. And yeah, so one more thing. The reason I was mentioning this, in urea cycle, this phosphate, so this PPI, breaks down on its own to make the like individual phosphates. So the 2P and P just breaks down on its own to make 2 separate phosphates. So PI like twice. So it just becomes P there. This reaction, the breaking down of PPI into its own molecules, uh, again, releases energy, you can say. Catalyzed by this enzyme, it releases energy basically. So now you have 3 ATP that broke down, plus this alone reaction that broke down PPI into P and P. So that's in total 4, right? So 3 ATP here, 1 reaction here. All of this in total makes 4 high energy phosphate bonds that were broken down. In general, this is what I was trying to explain this whole time. So 4 high energy bonds are used in when you make each molecule of urea in the urea cycle. Okay, so 3 from the original ATP breaking down, and 1 from the PPI that further broke down into P and P. Okay, I hope that was clear. Okay, I'll move on if that's okay. Wait, okay. Oh, wait, let me just clear this. So, yeah. So now we covered the overall reactions. We said 4 high energy bonds are broken down, 3 coming from ATP, 1 from the PPI. Now, urea, so again, remember how I said that all cycles are interconnected in some way or another? This is what happens to the urea cycle actually. So you might have heard of the citric acid cycle. It's called TCA cycle. It's called Krebs cycle. In first semester, it's a whole thing. It's the biggest cycle that everyone had to memorize. But yeah, it is connected. You don't have to know the entire cycle for this lecture, but just know 2 things are related to it. So remember how we said in the urea cycle, you have aspartate joining it, and then fumarate leaves it. You will see that multiple times. So where does it leave? Where does fumarate leave, right? Fumarate leaves, I mean, the urea cycle to join. It might sound familiar if you remember the Krebs cycle. It actually is a part of Krebs cycle. So I got this picture here. This is the Krebs cycle. One of the things in Krebs cycle is called fumarate. So the fumarate that comes from the urea cycle leaves and becomes a part of the TCAs or Krebs cycle. Okay, here, right here. Now, what is aspartate doing here, you might ask? So fumarate is part of Krebs cycle, right? It goes into malate, oxaloacetate. Again, you don't need to know the details of the Krebs cycle, but basically oxaloacetate, which is one of the, again, substances in the Krebs cycle or TCA cycle or citric acid cycle, it can get converted into aspartate. Okay, and aspartate, remember, it's the one in the urea cycle, the one that gives the nitrogen. So this is a picture. Again, no need to know it, but it's just showing you the convergence. Remember the transamination that I was talking about? Basically, this oxaloacetate has the ability to turn into an amino acid that is different. So aspartate is one of them. Okay. And again, you don't need to know how or the enzymes for this. Just know that there's transferring of amino groups to make aspartate. Okay, so in that way, we covered that the TCA cycle is part of the urea cycle. How? So wait, I'll start with this. So fumarate leaves, and it's part of the TCA cycle. And in the TCA cycle itself, oxaloacetate can or has the ability to turn by transamination, transferring things to aspartate. Aspartate can join in the third step, if you remember, third step to make the urea cycle derivative here. So the aspartate, this one, can come from Krebs cycle here and joining citrulline, and fumarate leaves this part to join the TCA cycle. So it's a mix and like it just comes and goes with the TCA cycle. Okay, so here it says fumarate is connected. I tried to mention all of that. Yeah, so oxaloacetate is the one that is transamination reaction to give you aspartate. And yeah, aspartate is important in the urea cycle. This is a very like a picture summarizing everything that I just said, plus all of these other substances. You don't really need to know it that much. Just know that aspartate and fumarate they're connecting the two together. Okay, fumarate leaves urea cycle to go here, and then aspartate comes from the TCA cycle to go into the urea cycle. They're both very important. Now, regulation of urea cycle. Okay, so what do you need for the urea cycle to take place? You need enzymes and you need the concentration or substrates, you can say. So these two will affect the urea cycle, as simple as that. So the enzyme levels will affect the urea cycle. If you have a lot of enzymes, you will have a faster urea cycle or more urea cycle. And if you have the substrates are high, if you have excess amino acids, all of these things, increase concentration, excess, anything like that, it will make the urea cycle faster or happen many more times. Okay, so enzyme level and substrate concentrations affect the urea cycle. All of these are the same. It depends on the diet of the person. If they're taking a lot of proteins, amino acid, like a protein-rich diet, and yeah. Okay, so the last point is like a side note, the last two. They're just saying that some amino acids, such as arginine and glutamate, can form the substrates or substances in the urea cycle. For example, arginine from the diet can make ornithine. Okay, ornithine is the carrier thing in the cycle. Glutamate, another amino acid, can be also converted to ornithine. So ornithine can come from within other amino acids such as arginine and glutamate. Okay, they can both form ornithine, which is again, ornithine is a very important part of our cycle here. Okay, it goes and recycles back. Okay, so it can be affected by diet, right? So it depends on your diet. how much of concentrated cycle or substances you have. Even the enzymes are affected, right? The enzyme level. Okay. So we said regulation of urea cycle, we said enzyme and substrates concentration. Now, the third thing, the rate limiting step. So do you see here? Find control. If they ask you which step is regulating the urea cycle, it's the first step, carboxyl phosphate synthetase 1. And regulation of this enzyme regulates the entire cycle. This is affected or controlled. Remember, like I said, it needs something to activate it, which is an acetyl glutamate. N-acetyl glutamate itself, so you'll notice here, this is the opposite reaction. So it's the same thing. Do you see this one? CP, carbonyl phosphate synthase 1 is here. This reaction is the same as this one. And N-acetyl glutamate is activating it. N-acetyl glutamate is activating this. Okay, it's just written in reverse. So N-acetyl glutamate itself is regulated by other things. So the rate limiting step needs N-acetyl glutamate. N-acetyl glutamate is itself regulated by two more things. Okay, so that's all going into the regulation of the urea cycle. Okay, what is it regulated by? N-acetyl glutamate comes from a protein-rich diet, like it was mentioned before, specifically glutamate and acetyl-CoA. Acetyl-CoA and glutamate, if someone has too much of these, they will have more of this, and that will regulate the urea cycle to keep going, right? So N-acetyl glutamate is dependent on these two substrates plus another amino acid, arginine. Arginine stimulates this conversion to N-acetyl glutamate. N-acetyl glutamate regulates this conversion from carbon dioxide and ammonium to the carbonyl phosphate. So this is the obligate activator of the first step. This itself is regulated by arginine and the amount of its substrates, glutamate and acetyl-CoA. So that's why it's called this acetyl-glutamate is called allosteric activator of this. So it is affected itself by other things on top of that, on top of it activating this, right? So it acts as a, okay, this is okay. I tried to mention all of this. And yeah, so again, N-acetyl glutamate, right? Its presence is determined by two things. The substrates, these two, glutamate and acetyl-CoA, if you have them, you will have this. And arginine. If arginine is there, it will stimulate. So arginine activates this conversion. Now, this conversion happens by the name again. It makes an acetyl glutamate, right? So the enzyme name is N-acetyl glutamate synthetase. Okay, it's not written in this arrow, but this enzyme makes this. It's activated by arginine, and it's determined by the concentration of substrates. And then this goes on to obligate, like it's an obligate activator of the first step anyway, as we mentioned. So it's very, very like regulated very well. Okay, so this is the last slide, I think, for this part, which is the urea cycle. We'll go into the diseases now in a bit. So again, this is another diagram. Everything was already explained in the last one. It's just another diagram for you. Just remember that there is no loss or gain because it's a cycle of no loss or gain of ornithine, citrulline, arginosuccinate, arginine. So all of these are like used up and they're made in the cycle anyway. So there's no loss or gain of this. This is all present in our bodies, in all of us normally, and they will not be used up or made in the cycle newly. What is used in the process is carbon dioxide, ATP, aspartate, and yeah, here, ammonium. So these, do you see carbon dioxide, ammonium, ATP? We've all mentioned this. Plus aspartate is used up in the cycle here, right? And yeah, the others, however, the one in the cycle that doesn't leave or, you know, leave or enter the cycle, these four, citrulline, arginosuccinate, arginine, and ornithine, are not gained or lost. They're just continuously in a cycle, and it's normally they're in normal levels. Okay. Okay, so this is the first checkpoint. I have two questions just to like recap on everything. You can pause and try to answer it on your own. So the first question is, which of the following represents the major compound of the circulation responsible for transport of nitrogen? So transport of nitrogen from the cells, right, across the blood, we said it needs to be safe, right? So is it alanine, asparagine, glutamate, glutamine, or uric acid? And I'll show the answer now. If you want to think about it, you can pause. So the answer is glutamine. Now, glutamine, we said, remember, glutamine is a non-toxic carrier of ammonia. It was in red. The reason that I put it here was just for you to like focus on this, and also not to confuse with glutamate. So glutamine comes from glutamate. Glutamine comes from glutamate, but glutamine is the ultimate like carrier across the body. Okay. I hope that's clear. This was in the first or second slide. Okay, question number 2. A 2-day-old infant is brought to the ER suffering a seizure. Analysis of serum ammonium reveals it to be quite high. Suspecting a urea cycle impairment, the ER physician, all of this is not that important, it's too advanced. But what they found here, after the hemodialysis, the infant is treated with arginine, which maintains the very high ammonium. The ability of arginine to render this effect stems from its role in activating the synthesis of an activator. So arginine activates an activator of the urea cycle. Which of the following represents this activator whose synthesis is induced by arginine? So you can pause it now if you want. Now, what they're trying to say is that arginine stimulates something which activates the urea cycle. Okay, I hope that's clear. So they're saying, what is that something? So as we saw in the last slide, the answer is N-acetyl glutamate. We said that, this is exactly from the last slide. So we said that N-acetyl glutamate here is an activator of the urea cycle, It is induced or affected, determined by the presence of arginine that acts on this, right? To make this in the first place. This will then go on to activate this. So N-acetyl glutamate is induced by arginine. That's what they mean from this question. It will not activate, arginine will not activate any of the rest, right? This is just to confuse you. It's always this one, N-acetyl glutamate activated by arginine. Okay, now I'll move on to deficiencies. So the cycle part is done, right? If you understand the enzymes that were involved, inshallah, this is easier. And yeah, so in this slide, you'll see the listed enzyme names. So you'll see carbamyl phosphate synthetase, ornithine, all of these we discussed before, synthetase, lyase, and so on, arginase, a lot of them. And this one is the last one, N-acetyl glutamate synthetase was right here, the last thing we mentioned here, okay? The one that was not in the arrow. Okay, so these are all enzymes. So there are many deficiencies, like diseases, which have deficient enzymes. So a baby can be born with any of these enzymes not working, and that's genetic deficiency of the enzyme. And the most common one is the second one. The most common one out of all of them are the first two, but the most common one out of these two is the second here, ornithine transcarbamylase, or OTC, is the most common disorder of urea deficiency. And these are genetic or inborn errors. They're from birth. Some people just have, like they might have mutation in the enzymes coding for them, and there are just not enough enzymes for the urea cycle to happen. So you can imagine what happens, right? If any of the enzymes are deficient or not working, you will have an increase or buildup of the toxic substance called ammonia. And like I said, ammonia is very, very toxic. You do not want that in your body. So here, okay, this table is talking about these first two enzymes. Oh, so if anyone has a question about this one, arginosuccinic acid lyase, it's the same thing as here. Wait, sorry, by the way, if I'm moving too fast. So this one, arginosuccinate lyase, is the same thing as saying arginosuccinase. So this is the fourth step. Arginosuccinase, I said it breaks down arginosuccinate. Sometimes you will hear that where the other name for it is arginosuccinate lyase. Okay, so it's just another name for it. Don't worry, it's nothing new. Okay, so this table is showing you deficiency in the first two enzymes. As we said, they're the most common, right? And this is more common out of all of them. Okay, what happens if you have less enzyme deficient enzymes? You will have, in both these cases, you will have high ammonia. It's building up, it's toxic because the urea cycle is not working. So that automatically means that uh, glutamine is high because we said glutamine is the carrier of ammonia. So if ammonia is high, glutamine is high in both of them. And so the first one is talking about the first step, enzyme is deficient. The second is second step, ornithine trans is deficient. Okay. What's BUN? BUN is blood urea nitrogen. So obviously, if the urea cycle is not working, the blood urea levels would be less in both of them. The urea is not being formed. Instead, ammonia and glutamine is accumulating and it's toxic. This toxicity, I'll skip over the blue for now. This toxicity is affecting the brain. It will affect the newborn. They will have very bad symptoms like lethargy, vomiting, and breathing very fast. And if not treated, it can cause, again, brain edema, fluid, and coma or death. Okay, so it's very dangerous. You do not want ammonia to be high or hyperammonemia. Okay. Now, what is the difference is the one in blue. How do you know if this baby has deficient in this enzyme or in this enzyme? You measure something called orotic acid. Okay. Now, I will explain how that happens, but basically, orotic acid or orotic aciduria is only high in ornithine carbamylase deficiency, not this one. Now how I like to remember it is like O and you know how the game like X and O or yeah the XO game. This one you can just memorize that O and X are related. So this enzyme ornithine transcarbamylase is related to orotic acid being high. In this one, there is no orotic acid. Okay. And the other thing, even here, oh, sorry, yeah, I meant O and X for this one. But O and O, ornithine transcarbamylase, and orotic acid, O and O, right? And then it is also X-linked recessive disease, genetic disease. So X and O is here. This one is, on the other hand, autosomal recessive, okay? So these two are the differences, key differences between these two enzyme deficiencies. So here it says orotic acid, uracil, all of these will be high in the second one, or ornithine transcarbamylase deficiency, O and O, but not in this, not in the first one. And yeah, so there you will hear orotic acid, uracil in pyrimidine, another cycle, you can say, or synthesis. Pyrimidine is in the DNA. You don't need to worry about that for now. But basically, these are intermediates, or these are used in that cycle. Okay, but these are just high in ornithine transcarbamylase deficiency. If someone doesn't have this enzyme, they will be high. Okay, so what they're saying here is the blue thing. So if you have decrease in the second enzyme, this one, you will have increase. I will explain this right now, but you will have increase in buildup of the previous product, which is this one. So if this one is deficient, if you have an X on this one, that means that this will build up, right? even ornithine. Both of these cannot go to citrulline, so this will build up. Carbamyl phosphate will be high in the blood, which will increase the pyrimidine pathway because it's a part of that. So pyrimidine pathway will increase, which increases uric acid, uric acid, and uracil. Okay, so this is what they mean. The CUT are the pyrimidine, cytosine, uracil, and thymine, if I'm not wrong. So this is all pyrimidine pathway, and in the pyrimidine pathway, you also have orotic acid in that cycle. So that's why it's high here. Okay, because carbamyl phosphate is related to pyrimidine pathway, and it goes into that direction. Okay, so I just brought this to explain that the deficiency. So obviously, if you have deficiency here, you will have buildup of like the back products, so carbamyl phosphate or ornithine, and you will not have urea or citrulline or all of these. Now, if you have deficiency in the first one, which is carbamyl phosphate 1, this one, you will not even have carbamyl phosphate. So the cycle will not take place, right? Everything here will be low. Okay, urea will be low. Ammonium will build up actually instead. Okay. Now, this is what we said. The first two enzymes are the most common deficiency, I mean, and OTC is the most common. It can result in really bad symptoms and increase in orotic acid. Okay, these two, I'll try to explain them in a bit, but basically, if you have in any case, in these two cases, you have hyperammonemia, right? So this excess buildup will also cause buildup of the amino acids such as glycine, glutamine, and alanine. So glycine, glutamine obviously will build up as it's the carrier. Glycine will also build up because it's an amino acid in excess. So glycine, glutamine build up because yeah, so pyruvate, just know that you don't need to know the details of it, but pyruvate glutamate combine with the excess hyperammonemia to make these two substances or these two amino acids. Alanine is another amino acid that increases. So for you, just know that glycine, glutamine, alanine are the three amino acids that are high if someone has urea cycle defect. Okay, it's just buildup of ammonia. That's why, and you have back of um, amino acids. Okay, backup of amino acids. Now, this is the first one. Carbamyl phosphate synthase 1 deficiency. So the first enzyme is not there. The first step will not happen. The rate limiting step will not happen. And so this is the autosomal recessive one. Okay, so these are the names, and I put arrows, like I'll go through them one by one so that it's clear. So carbamyl phosphate synthetase 1 deficient, right? It's deficient. So obviously it will be less. This is what we're talking about anyway. Citrulline, which is this product, if this is not happening, then this will not happen and this will not happen. So this is less and this is less because it's not going forward, right? What about orotic acid? I said, or we said, that orotic acid doesn't increase in the first enzyme, okay? Because it only decreases in the, or increases in the O and O. So this will be less still now. What about ammonia? So we said if this enzyme is deficient, ammonia will back up, it will build up in your body. It's very toxic. You don't want that for the baby or anyone. So ammonia will be high. That's the only thing that will be high in this deficiency, the first enzyme. Now, second deficiency or second enzyme in the urea cycle, the OTC, if that is not there, this one, okay, it makes citrulline, right? Usually it combines with this one to make citrulline. If that is not there, then citrulline will not happen in the first place, and the backup of this reaction will be higher, right? Will be higher. So I'm just want you to relate to this. So if this enzyme is deficient, citrulline will be down, ornithine will go up, or this one actually is the backup reaction. So carbamyl phosphate will go up, because it's not able to go forward. Okay, so that's what I mean. So ornithine transcarbamylase, obviously we're talking about the enzyme OTC deficiency, so that will be less. Citrulline will be less because it's not going forward. And carbamyl phosphate, the backup will be high, very, very high. It cannot go forward because there is no enzyme. What about orotic acid? So we said carbamyl phosphate is related to pyrimidine pathway. Pyrimidine pathway consists of something called orotic acid. So O and O. So this buildup shifts into the other cycles for the pyrimidine synthesis, basically increasing orotic acid. Okay, because that's all a part of that cycle. So carbamyl phosphate buildup, which will go into orotic acid, make more of that, and it will both increase. What about ammonia? So ammonia, we said that again, if you have no urea forming, ammonia will build up anyway. Like if you have carbamyl phosphate building up, you will have excess ammonia. It cannot go forever into like forward into this. It will also build up. So all the back reactions will build up, ammonia will be high. So in the second OTC deficiency, second enzyme, you have increased carbamyl phosphate, backup of orotic acid, and backup of ammonia, backing up, I mean, okay? The deficient will be the forward reactions with the enzyme also, obviously will be deficient. And remember the X and O. So this one is X-linked recessive. The previous one was autosomal recessive. Okay. So these two enzymes are deficient. They present differently with different levels. So if you measure that in a baby, that's how you know that this person has, which enzyme is deficient in that baby. Okay, we're almost done. Um, Remember how I said that glycine, glutamine, alanine are all very high in urea cycle defects? You can give something to remove these high uh, amino acids or excess amino acids. So for example, if you give benzoate, if you feed the baby benzoate, it will combine with excess glycine to give you something called hippurate. This is safely excreted, okay? And it can be excreted. Now, what about glutamine, which is another buildup? If you give phenylacetate, it combines with the excess glutamine to remove it as phenylacetyl glutamine safely. The longer, you can say the substrate, like substance that you give to the baby, the longer name, it's with the longer name. The shorter one, benzoate, is hippurate, okay? So this is shorter, this is longer names. Basically, they just convert the excess amino acids into safely excreted products. So you can give them benzoate, phenylacetate to remove this. Another thing you can do is just not feed or try to restrict the protein diet in that baby or person. So if you reduce protein intake, they will not have buildup of ammonia that much, right? So if this is not happening, the urea cycle is not happening, there you have buildup of ammonia anyway, and you don't want to put more protein into the body. So another solution other than these is to restrict that protein so that this doesn't build up more. Okay. Yeah, can be treated with a low protein diet. Now, another deficient enzyme that can happen is this one, arginosuccinase deficiency, which is this one, the fourth reaction. Arginosuccinase, if there's a deficiency here, this part will not happen. So you will not have fumarate, will not have arginine. This will build up because it's not going forward. This will build up and this will be secreted in the urine instead of urea. You will not have urea because it's not going forward. This will instead be excreted. Treatment for this, you can give them, again, same thing, low protein diet because you do not want buildup of ammonia by giving them too much protein. And another thing, you can give them arginine to get rid of the ammonia by stimulating the urea cycle. So there's another picture for it here, I think. Yeah, here. So this is the same thing. So they're saying low protein diet and high in arginine. Low protein, everything else, but that's because you don't want buildup of ammonia too much. You want it to be restricted. And in case of arginosuccinate deficiency, I said that this will be excreted. You will not have urea. You can give arginine to kind of stimulate this cycle, because remember, stimulates, arginine stimulates N-acetylglutamine, and acetylglutamine stimulates this first step. So it will kind of get rid of any excess ammonia to go to the arginosuccinate. So arginine, you can feed them to increase the rate of the cycle, and it can be excreted as this, okay? It will just not go to this step. So you are giving them arginine. I think this is, I hope that is clear. Just remember that line that you give low protein diet and high arginine, you can give that. Because this step is not happening, you want to give arginine, right? And yeah, this is basically excluded. Now, one slide that I skipped here is, it's basically the same thing. If you have urea cycle defect, you will have toxic levels of ammonia or ammonium and excess glutamine, because it's a carrier of that ammonia. So glutamate, this is from the first slide. So glutamate combines with ammonium to give glutamine with glutamine synthetase, right? This is the first slide with the muscle picture. So this is in places other than the liver. Basic idea, what I'm trying to say is that if you have too much ammonia because of this urea cycle defect, you will have excess glutamine as well. Okay. So yeah, this is very, very dangerous for the cerebrospinal fluid in your brain, and you don't want too much of the glutamine coming from the too much ammonia or hyperammonemia. Now, all of this very high glutamine will lead to brain damage, and that's what you don't want. So you give them low protein diet with high in arginine to kind of fix this cycle or this defect. Okay. This is the third enzyme that can be deficient, as we said, this can be deficient. But the most common is, again, OTC deficiency. Okay, this way we're done. Inshallah, it was okay. I have a few questions, we can answer them, and you can pause and answer them if you want, and I'll try to explain them. So, a newborn becomes lethargic and drowsy 24 hours after birth. Blood analysis shows hyperammonemia coupled with orotic aciduria. This individual has an enzyme deficiency that leads to inability to form which of the following. So you can pause it if you want. If you have hyperammonemia with orotic aciduria, that means you have deficiency in OTC, right? So I'll explain. So you have deficiency in this one, OTC. It's now that makes it inability, like that makes it unable to produce which of the following forward reactions? This one, citrulline. So OTC doesn't make ornithine, doesn't make this one, it makes citrulline. So if you have deficiency in this enzyme, you will not be able to make the forward reaction, which is citrulline. If you see orotic aciduria, hyperammonemia, just know that this is OTC deficient. It is not there enough by genetic diseases, like we mentioned, right? So unable to um, create more citrulline. That's what they mean. Okay, question 4. A female neonate did well until 24 hours when she became lethargic. You can skip that. At 56 hours, she started showing seizure activity. And when they measured the ammonia level, it was found to be very, very, very high. So hyperammonemia. Quantitative plasma amino acid levels revealed marked elevation of citrulline, but not arginosuccinate and urea. Which one of the following enzymes is deficient? Now you have to find which enzyme is deficient in this baby, right? Because you can pause it now if you want. You have marked increase in citrulline, but not arginosuccinate. So I'm trying to find a picture. So if you have increased citrulline and not enough arginosuccinate, not enough urea, then that means where is the block, the enzyme? It's right here. Okay, this enzyme is not working which means this is less and that is more. That's what the question was saying. So what is that enzyme? So you have increased citrulline, but not the forward reaction, arginosuccinate. So the enzyme that was in that step is called arginosuccinate synthetase, making of arginosuccinate, right? So if you don't have this enzyme, you will not have the forward reaction, which is making of that. Okay, so that's why it's deficient. Okay, which of the following would be elevated in the blood of this patient? This one. So if you have arginosuccinate synthetase deficiency, which is a urea cycle defect, what will be increased? Is it asparagine, glutamine, lysine, urea? Which of the amino acids or substance will be increased? So we mentioned glycine, glutamine, and alanine will be high, right? So from here, glutamine being the carrier of ammonia will be high, right? So here, this is just another picture to remind you that we are here. Arginosuccinate synthetase is not there. It's not going forward. Citrulline is high. And this builds up the ammonia. This baby is has very high ammonia or ammonium. And that's why she will also have high levels of glutamine, which is the non-toxic carrier of ammonia anyway. Okay. Okay, two more questions. Question 6, an infant born to a 32-year-old female demonstrates lethargy, vomiting, seizure, same symptoms, you see? So they have hyperammonemia, as we said. So they have increased blood ammonium level. Liver biopsy suggests impaired formation of N-acetyl glutamate as the cause of the child's problem. Which of the following is impaired? Okay. So which of the following as in A, B, C, D, or E? Okay. So if you don't have, you can think about it if you want, you can pause it. If you don't have N-acetyl glutamate, which one of the following reactions is not happening? So we said N-acetyl glutamate is an obligate activator of the first reaction, which is A. So first reaction N-acetyl glutamate is not there. So the first reaction will not happen. It is impaired. Okay, from the beginning, because this is the rate limiting step and you need this to continue. This is just another cycle in this format, so don't worry. Okay, last question. Which enzymes are responsible for producing the direct donors of nitrogen in urea? So we said urea has two nitrogen, right? One, I'll answer it now. One from aspartate and one from ammonia, right? So where does it come from? Does it come from arginase and arginosuccinate lyase? These are the last enzymes in the urea cycle, so not these. Xanthine oxidase, we didn't mention that here. Glutamate dehydrogenase, glutaminase, it was not mentioned here. Plus, there's nothing that says aspartate or ammonia, right? Now, D, arginosuccinate synthetase, ornithine transcarbamylase, or aspartate aminotransferase carbamylase. Okay, you can try to answer this if you want, but basically what the idea of this question is, where does the ammonium come from and where does the aspartate come from? So E says aspartate aminotransferase, so that's one. Aspartate gives one nitrogen. And carbamyl phosphate synthetase is the first enzyme in the urea cycle. It makes, it integrates ammonia, right? Ammonium, right? So that's why this is the second nitrogen that goes. So wait, I hope I put a picture. Okay, I didn't. Okay, here. So the first, so urea has two nitrogen, right? Two nitrogen. One of them comes from ammonia or ammonium through this enzyme, which incorporates it into the urea. And the second one comes from aspartate, which can be from aspartate aminotransferase. Remember the amination reaction from Krebs cycle? So if you see aspartate, that's your answer. Okay, aspartate aminotransferase. Even it could also be this one, arginosuccinate synthetase. but that's a trick question because it's not ornithine transcarbamate. Okay, so here, this one, I mean, it does, but it's more correct to say, like, it goes into urea eventually, but it's more correct to say this one because it directly incorporates the ammonium. Okay, so that's one nitrogen down. The second one is from aspartate. So aspartate, trans amino reaction from the Krebs cycle gives you the aspartate directly. So that's the second source of nitrogen for urea. I hope that was clear. If not, I wrote the explanation in under each question, I believe. And yeah, if you have any questions, feel free to reach out to me through my email. I know this was the recording, but yeah, I'm available all the time. This is just a picture for you to remember that the mnemonic orange colored cats always ask for awesome umbrellas. Thank you for listening. I hope that was all good. And best of luck in your upcoming exams and in MOL in general. Thank you.